Welcome back with another video lecture, Malaria. Let's start. Malaria is an infection which is not caused by a mosquito, but malaria is caused by a parasite Plasmodium that a mosquito carries and which belongs to protozoan genus called Plasmodium. So the mosquito in this case specifically called female anopheles, and they carry a parasite Plasmodium. The malarial infection is very serious problem, especially in underdeveloping and tropical countries, because there are 300 to 500 million malarial cases reported annually, and which 2 to 3 million death rate per year, mainly children. The Plasmodium parasite belongs to Phylum Apicomplexa. Those parasites which have complicated life cycle are placed in Apicomplexa, and malaria is one of them. There are 5 Plasmodium species that affect humans. In all these types, Plasmodium Vivax and Plasmodium Falciparum are the main causative agent of malaria. The Plasmodium Falciparum are the more severe form of malaria and found in Africa. Plasmodium Vivax are mostly found in Asian countries. Plasmodium Ovale are found in Asia as well as South America. Plasmodium Malariae are found in Africa, and Plasmodium knowlesi are found in East and South Asia most commonly. Now the life cycle of malaria. The life cycle starts when the female mosquito bites human and sucks blood. As a result, it injects sporozoites to human body. The sporozoite is a term that is used for immature form of Plasmodium parasite. So the sporozoites inject into the bloodstream and within 30 to 50 minutes reach to hepatocytes or liver cells and take up residence there up to 6 or 7 days. During this stay period, the sporozoites start multiplication asexually to form so many merozoites. If there are so many merozoites in hepatocytes, it is collectively known as schizont or schizont. All these merozoites are then released by the liver cells and back into the bloodstream by rupturing hepatocytes or liver cells. This multiplication are called pre-erythrocytic cycle or exoerythrocytic cycle because they cannot infect red blood cells directly. This can take 7 to 10 days, while some species of the schizont, that is Plasmodium vivax and Plasmodium ovale, make hypnozoites due to improper dose of medication or some other reasons. They go to a dormant hepatic phase called hypnozoites. The hypnozoites don't divide, but they live in dormant form and causing a long delay between the initial infection and symptoms from the disease. This liver stage or exoerythrocytic stage is generally asymptomatic. When all these merozoites releases into the bloodstream, they invading red blood cells and multiplying again to form much more merozoites until RBCs burst and again release merozoites into the bloodstream. So the merozoite will infect this particular RBCs and will make a ring-like structure or immature trophozoite. Then the trophozoite, it will multiply to form so many merozoites or mature trophozoites, and then schizont or schizont. At last RBCs will burst and releasing merozoites and get back into the bloodstream. So what will happen here to all these merozoites? The cycle will just repeat it. The merozoites will invade more RBCs, they will multiply and burst. Each time this will occur, which will cause chill and fever and also sweating, which are the typical symptoms of malaria. After several sexual and asexual cycles, the merozoites can infect RBCs, instead of replication and multiplying, they will develop into sexual form of Plasmodium parasite, which are Plasmodium gametocytes. Now they are of two types, that is male gametes and female gametes, or macrogametes or microgametes. So here what these gametocytes will do, will they want to do something else in human body? They require female mosquitoes or anopheles mosquitoes in order to become a sporozoite and infective form of Plasmodium species again. So therefore, if another uninfected anopheles mosquito come along a female and bite this particular infected human, it will suck up these gametocytes, the male and female gametes. So the mosquito will bite this infected human, it will digest the male and female gametocytes. Now the male and female gametocytes are fused to each other, forming zygote, which are highly motile, and we called it ookinete. The ookinete will form an oocytes. Oocytes are those forms of parasite from which sporozoites begin to develop. These sporozoites will multiply and then will cause the oocyte to rupture, releasing the sporozoites. It will move into the salivary glands of a mosquito, ready to be injected into another human being. So again the male and female gametes enter the sporogenic cycle, producing more pathogenic sporozoites. The mosquito in this case can then bite another human and infected, causing malaria, and this cycle will repeat again and again. Here are some symptoms of malaria include headache, fatigue or tiredness, nausea, vomiting, shaking, chill, fever, muscle aches may also occur. Malaria also may cause anemia and jaundice, which is yellow coloring of the skin and eyes because of loss of red blood cells. In many cases of malaria, the fever and chills occurs in episodes or in interval. For Plasmodium Falciparum, the fever is variable, sometimes just like tertian fever and sometimes daily within 24 hours. That's why called malignant tertian fever. In Plasmodium Vivax and Plasmodium Ovale, fever happens every 48 hours, called tertian fever. For Plasmodium malariae, fever happens every 72 hours, called quartan fever. This cyclic pattern of fever is due to rupturing of red blood cells and releasing of parasite in cyclic manner. Pathological changes occur in various organs in malaria. Number 1 is splenomegaly or enlarged spleen due to high phagocytic activity of erythrocyte and macrophages. Hepatomegaly or enlargement of liver because of Kupffer cells are increased in number, and pigment seen in Kupffer cells and parenchymal cells. The bone marrow can become red and hyperplastic in malaria. In malaria, kidney become enlarged and congest. They can infect heart, that is coronary occlusion or thrombosis. Pigments are present in adrenal glands, lungs and stomach. Brain congest because of malaria. The malaria diagnosis can be done through microscopy commonly. This method is used for demonstration of parasite in blood, usually by thick and thin smear, where thick smear is for parasitemia or just for parasite detection, while thin smear for malarial species identification. Here are the morphology of some species in different stages, that is ring form, schizoid form and gametocyte forms. The morphology of Plasmodium falciparum have small ring and double chromatin dot. The schizoid of Plasmodium falciparum having 12 to 30 merozoites in each RBCs, and the gametocytes of Plasmodium falciparum are kidney or banana shaped. The ring form of Plasmodium Vivax have large ring as compared to Plasmodium falciparum and have a single chromatin dot. The schizoid form of Plasmodium Vivax have 12 to 24 merozoites, while their gametocytes are large and spherical. Plasmodium malariae have small compact ring and single chromatin dot. Their mature schizoid have 6 to 12 merozoites. Their mature gametocytes are round. Plasmodium ovale have large and compact ring having a large dot. Mature schizoid of Plasmodium ovale have 4 to 12 merozoites which are scattered, and their gametocytes are spherical in shape. Here are some prevention of malaria. Number 1. Drape mosquito netting over bed. Drain and close the window. Use insect repellent and use mosquito repellent spray. Also reduce the risk of mosquito bite. So how will you treat malaria? We can treat malaria with anti-malarial drugs like chloroquine, lumefantrine, pyrimethamine, chloroquine and amiodarone. We can also use supportive therapy for malaria. For severe blood loss anemia, blood transfusion can help. Antipyretic for fever and fluid and electrolyte balance to prevent fluid overload. Thanks for watching.